I'm Janet Pregler. I'm the director of the Iris Cantor UCLA Women's Health Center. And I'm really excited to be here with some of the leading innovators in women's health and technology at UCLA. I'm going to start this with a story. So when I was in medical school, I learned about the 70-kilogram man. Is there anybody here who's heard of that? Okay, that's kind of good. But we learned about the 70-kilogram man. We learned about the heart and the liver and the kidney in the 70-kilogram man. And in the 80s, and in some ways even until today, when we think about women, we were told it was a smaller man plus a little black box that had the reproductive organs in it. So we really learned about 70-kilogram men and 60-kilogram men. And I still remember when I started talking about women's health with the then vice chancellor for research at UCLA, who was a physicist. And he said to me, I'm a physicist, but I definitely can see that there are flaws in that model. And that was 100% true. So what's the impact of this been on research, and why is this an opportunity now in research? The male model really led to funding for male, primarily conditions, with a few exceptions where there was strong advocacy, like breast cancer. And even in 2021, when we looked at National Institutes of Health funding for different diseases and conditions, and we compared the amount of funding to the quality years lost for condition, out of the 15 top conditions where there was a mismatch in funding, too little funding related to the importance, 14 are primarily in women. So I think that shows you that this is really a profound issue. Now, Dr. Carol Watson, who's on the end here, she's an internationally renowned cardiologist in women's cardiology, as well as cardiology in general. Was present with President Biden in March of 2024 when he announced a new initiative on women's health research that dedicated basically $1 billion in fiscal year 2025 to women's health research through a variety of funding mechanisms, a prioritization of expanding that research, as an example, through the National Science Foundation to include engineering and computer science. And also asking the Office of Management and Budget to really look at where are things in terms of statutes, in terms of policies and procedures that could be changed to promote women's health research. So now I'm going to turn it over to just this incredibly wonderful group of investigators who are doing work that intersects with the theme of LA Best today. And I'm going to start with Dr. Lenny Ackerman.
All right, so I'm a urogynecologist, a clinical urogynecologist and researcher. And what that basically means is that I take care of what are called functional urologic disorders. And those are things like incontinence and urinary frequency and urgency and prolapse and getting up at night to go to the bathroom. And while these are non-cancerous conditions that people may write off, they actually are highly impactful and highly prevalent. And we don't talk about them, but I promise you, if you look around the room, that at least a third to a half of the people in this room probably have one of those symptoms to a level that's actually significantly impacting their quality of life. And that number goes way up as we age, so that, by the time we're 80, it's pretty much everybody. So if you live long enough, you're going to end up with one of these diseases, but they are both highly prevalent and highly impactful, particularly in our older population, where the stigma and the shame and the social isolation that are associated with these conditions can have a psychosocial impact on their quality of life that actually exceeds that of being stuck in renal dialysis 3 days a week. And so this is really obviously important, but I'm here to tell you that we do a really terrible job of taking care of these. And I'd like to think that that doesn't mean that I'm a terrible doctor, but we just really do a bad job. And part of that, it's multifactorial, but part of that is that only about 10% of the people with these conditions actually will ever see a physician for it. But worse than that, of those people, only about 10% of those will actually find any kind of effective therapy that they remain on long term. And so why do we do such a terrible job with these? Well, I would like to convince you today that I think part of that reason is the way that we approach these conditions. And the way that we do it is we have a couple of different diagnoses, and they're things like overactive bladder and interstitial cystitis that are basically based on the recognition of a single dominant symptom. So urgency and urge incontinence for overactive bladder, for example. And what that ends up meaning is that these conditions actually are completely arbitrary. In fact, for the most part, they were actually generated by pharmaceutical companies. No offense to pharmaceutical companies, this is great, but they were pretty much just arbitrarily given names so that the FDA would have something to pair up drugs with an indication. The diseases themselves, or these conditions, actually have no biomarkers, no tests, and frankly, no understanding at all of the underlying pathophysiology. In fact, if you ask most urologists, we will all pretty much agree that probably each of these conditions represents a collection of different conditions, with each with its own pathophysiology. So 5, 6, 7 different diseases that, while they all manifest with the same end symptoms, they actually probably have a number of different reasons why they happen. Each of those different reasons probably needs a slightly different treatment, which I think explains why we do such a terrible job. And so maybe we could approach it differently and think about symptoms. Well, the problem there is that really in the symptom world, pretty much everybody will present with more than one symptom. They almost all overlap. And the only real difference is kind of in the ratios in which they occur. And so that's really led to a lot of diagnostic confusion. It ends up with people not getting therapies that are effective and really being hopeless and ending up, sitting in diapers for the rest of their life, which is a terrible outcome that we want to do better. But that also is a wonderful place to start if you're thinking about using artificial intelligence. And that's basically where we went. And so what my research group has started to do is to use artificial intelligence or machine learning algorithms to try to change how we approach the diagnosis of urinary diseases. And so what we've done is we've taken large populations of care-seeking women and taken all of their symptoms, just asked them what bothers you in a bunch of validated questionnaires, and fed that into a number of different algorithmic, classification schemes. And in these, we're basically asking the computer algorithms to classify patients based entirely on the information coming directly from the patients, and we provide no input at all in terms of how to do that classification. We're asking the computer to see the structure in the data. And what we basically found is that instead of that 4 different conditions or 5 different conditions, what we've ended up doing instead is we now come up with about 9 or 10. And we've tested 9 and 10 differently. They're both kind of good, but 9 ends up being actually the best one. What we now find is that instead of classifying people based on this one dominant symptom and trying to decide that that's the right thing, instead we're now classifying patients based on those combinations of symptoms that are really hard for people to recognize, but the computer is actually really good at recognizing. And so just to try to speed through this a little bit, what that ends up looking like, and this is my sort of like trying to explain this in a 2-dimensional space, is that you take all of this information from patient symptoms and you collapse it down into 2 dimensions so that each individual patient, which is like a little spot on my little graph there, the people that have similar symptoms are close to each other. The people that have really different symptoms are far away from each other. And so our 9 different diagnoses now actually make a ton of sense. And you find that our pain patients are actually kind of clustered on one side of that big blob. And they kind of are similar in space, but now they start to segregate. And so we find that our pain patients that used to all be classified as one different disease that basically was defined as pain in the pelvis when you can't find an etiology, which is a terrible definition for disease. Now we finally have 3 different diagnoses for pelvic pain that each have very, very different and specific features. And when we start to look at how they respond to treatment, now those different categories actually segregate really well with how they respond to treatment. So we're already doing better. And the other really neat thing is that what's come out of this is that we have several diagnoses that didn't exist before. And so these patients who would come in and we would be like, well, I don't know what's wrong with them. They don't really fit anything. Now they have a home. Now they have an actual diagnosis. And what's really interesting is that those patients actually, they're kind of falling into disease categories that we all kind of knew were there, but nobody had really called them out. But now we have a way to actually recognize them. So what do we do with this information? So now we have this new diagnostic system that I hope I've convinced you is better than what we were dealing with before. What do we do with it? Well, what we did was to train an algorithm to actually take those patient-reported symptoms and place patients into these categories to provide some assistance to physicians. And what you can maybe see in the background is that there's sort of the big dots, that's our training set, but there's some little dots in the background. And we have since validated this approach, this algorithm, on 3 different populations of people. We've got a data set from another academic institution. We've got a big nationwide data set from one of the NIH research networks. And then we actually have a population that we recruited from all over the US who had never sought care for their urinary conditions at all. And in each of those different situations, we find that our classification is highly, highly consistent across these different groups. So it works really well to classify patients. But does it work for care? Can it work better for actual clinical management? And we find that it actually works really pretty well, but I'm gonna try to convince you of something, which is that when we compare the diagnostic algorithm that we created against physician diagnoses, and these are diagnoses coming from, super subspecialist physicians, dual board certified, urogynecologists, the diagnoses line up about 70% of the time. And that seems okay. I would love to be able to say that it's a little better than 70%, but I'm going to try to convince you that it is a lot better than that. And that probably is underestimating greatly the actual efficacy of this algorithm. Because when we start to look at treatment assignments, we find the algorithm does significantly better. And I think what that tells me, and I hope it tells you, is that when patients are coming in, some of these specialist providers are really recognizing that people don't fit in those categories, and they're assigning treatments differently accordingly. And so we're actually closer to that subspecialist seeing those patterns that maybe your generalist doesn't see than they are to actually using the diagnoses. And then the other thing I mentioned was that some of these diagnoses don't exist, right? We're finding some new diagnoses. And so obviously that accounts for some of the mismatch. But there's one other thing that I think is super important, which is that these are really, really shameful conditions for a lot of women. And I wish I could point out, but somewhere up in the top, there's a red bar that looks up like that. And that is your population with fecal incontinence. And it turned out that not a single woman in that category had told their physician, or maybe their physician never asked. I don't know, but 100% of them were missed. But we found them with the algorithm, which means we could do a better job of taking care of them. And so where are we going with this? Well, our hope is eventually, and this is what we're working on now, to develop a provider-facing clinical decision support tool where the patient can log on to some HIPAA secure portal, fill out their little questionnaires, talks about their symptoms, and that can provide a report to physicians or to providers of all kinds, from the generalist and the NP to the specialist, that can give them some advice on what to think about in terms of the diagnosis of the patient and what might be a useful treatment for them to start off with, hopefully making it more effective for people to get care, recognizing these conditions that can be very impactful earlier, and doing a better job overall of taking care of women. Thanks.
Thanks so much, Lenny. Now we're going to hear from Dr. Yalda Afshar, who is using a chip technology to address a major cause of maternal death.
We create people, like literally humans create people. 2 cells come together and create a fetus. And that's what I study. I create the creation of an embryo and the placenta, which is an organ that you just create and then you dispose of at birth. It should be humbling and mind-blowing, literally looking at every single one of us. Yet we have such little understanding of the basic biology behind pregnancy. In the United States every year, there are 4 million births, 500,000 in California. The vast majority are fine, but a lot are not. Of those births, of those 4 million births, about 1/3 of them are cesarean births. And in red, all you need to know, that's the rate of cesarean birth in the United States. It is going up. With that increasing rate of cesarean birth, a disease that we've created called placenta accreta spectrum, or accreta, has arose. It affects about 1 in 300 births in the US. So that's when that placenta, that organ that was created, the afterbirth, doesn't detach from the uterus, and it can cause severe bleeding, hemorrhage, really sickness, ICU, transfusions, and even death. And we know the staggering reality that has been in the news of the increase in both maternal morbidity, or really, really bad sickness, morbidity as well as mortality. So because of this trend, we were seeking to do something about it. What can we do is early detection. So know when it's there, because when we don't know it when it's there, that's when things get pretty bad. So liquid biopsies, you guys are in tech, you know all about cancer, had really good advertising, right? Like studying tumors in the blood, using a marker to look in the blood. And really Mother Nature did that experiment. If you think of pregnancy, it's kind of like a cancer. 2 cells come together, rapidly divide, create organs, all these immune modulators that I was at the end of the VC. I mean, pregnancy, you have external DNA and your body doesn't reject that. So Mother Nature has done the experiment, and that's why I really hope we can think about pregnancy in that way, but it does it so correctly. So one of the things we've done is looking at markers of the placenta, that organ that was created in pregnancy, in the circulation of the pregnant person. This is a collaboration with a couple labs at UCLA who are based in CNSI, Sang and Ju, and our group. I really came to the table and said, let's study pregnancy. They're an amazing liquid biopsy group. And what they've done is created nanochip technology that's been published, patented through TDG at UCLA. And in essence, it's a rare cell sorter. And if someone who can click away, that's a little movie. So from just a normal blood draw, we're able to isolate some rare cells. The vast majority are just boring red blood cells without much information, but we're actually interested in those placental cells that we can get from circulation. And what we've done is we've utilized this as a early detection. So with that small blood draw, really early in pregnancy, now we do you have Down syndrome, not, what's the sex of the baby? They know by 9 weeks. At around the same time, we can utilize this to say, what is the status of your placenta? Is it an accreta or is it not? So that's great for screening. That's great for decreasing morbidity, but what is going on? And if we can use that biopsy as like a way to look into that placenta during pregnancy, and that's been a lot of the goal of our lab, is to think about what is the biology that underlines the placenta in the normal case to understand the abnormal case. So we use some really cool spectroscopy with, again, colleagues from the Advanced Light System at UCLA. We have the appreciation of patients that trust us for their care here for this condition, who donate and give us biopsies of their samples. And then we study it. What does the cell look like? You can just pretty much see purple's different on both sides. That's the point I'm trying to make. In one case, purple's at the top. In another one, it's interdigitated throughout the placenta. Clearly, they're different. And we can actually see that to think about what we can do differently surgically to then prevent this. And we've done this, that's another video. If I don't know who I'm talking, but a little click would be great, just to show that we can see this in 3D. So we do this by ultrasound and we can understand the collagen. So the matrix, it's such an easy intervention that we can do to hopefully prevent it. And that's really what we're trying to do. So number one, we're trying to identify an early biomarker for accreta. And I'm just using accreta as an example. It's one that's very close to my heart and I think about, but you can use this to look into the placenta during pregnancy. It could be about dating. It could be, we can talk more about all the things it can be, but an early surrogate biomarker. But really, really what keeps me up at night and what we need to push for is rethinking what is the biology? Why does Mother Nature do it right most of the time? And I think if we understand that, we can get clues to really then improve the care and intervene when we need to. And overall, the goal is just to improve the care of the pregnant people and then their neonates during the pregnancy and then throughout. But I really am going to push. It's, the individual cells, what cells are talking to who, because that's where the therapeutics, that's where the prevention strategies come from understanding that biology. And then as the theme is all research, none of this is done in a silo. This is multiple teams from engineers to biologists, clinicians, and the whole mark. But we have an awesome, diverse team with really, really different ways of thinking and approaching this. So pregnancy is where it's at. A normal physiologic state that we have no understanding of, but can give us clues to adverse both pregnancy outcomes and other diseases.
All right, so as you can tell, we have entered the black box, and now we will do more with the black box. Dr. Amanda Clark.
Thank you, Janet. So my name's Amanda Clark, and I'm the director of the Center for Reproductive Science, Health, and Education at UCLA. This is a new center at UCLA that was created in order to bring together biomedical researchers who work at the interface of innovations in reproductive science and health, and also to partner this innovation with the social sciences so that we can better understand how these new technologies in reproductive health live and are accepted out into the world. What Janet has asked me to talk about today is my own research, which is occurring in my lab. So we believe that reproductive health is women's health. And the organ that my lab specifically works on is the ovary. The ovary is unique to the female body. The ovary is not found in the male body. And when I say male and female, I'm referring to babies that are assigned female at birth. And from now on, I'm going to use the word female and women. So our goals are to understand the development and the differentiation of ovarian cells. And we do this because we're interested in creating in vitro cellular platforms using stem cells for research innovation in order to better understand disease, dysfunction, and non-function of the ovary. Our ultimate goal is to provide women with new options beyond what they currently have. And what they have is not much. In order to improve the function of the ovary, what they have is hormone therapy. They have aspirin. You can have your ovaries removed, so oophorectomy, ovarian stimulation and IVF if you're having trouble having a baby, or hormone-based contraception. And on that last point, what I want to say is that 30% of women are unhappy with their contraception because they're unhappy with how the hormones make them feel during their reproductive years. So this is a really interesting part of the marketplace, which is to identify new non-hormonal contraceptives, because contraception is something that lives out there in society and that women desire and want so they can control the spacing and the number of children in their family. So creating increased options for these women is really important. And that's also a part of the work that we do as well, focusing on the ovary. So I've had a lab at UCLA now for 18 years. I started here as an assistant professor. It was my first position out of my postdoc, which is at UCSF. And in coming to UCLA, I was recruited here as a stem cell scientist. So my lab is using stem cell models to build the ovary. But as we started working with stem cells in the lab, we realized if we're making cells in the lab, we'd better understand how these cells are first made in the body. So when we're making cells in the lab, we know that these cells recapitulate the sorts of cells that are found in the body and would function normally. So the ovary project was born, and it was born about 15 years ago. It began with initial collaborations between my lab and Ian Glass's group in the Human Birth Defects Lab at the University of Washington. We then built in new collaborations with Brad Kansas' lab at the University of Utah as we started to move into more genomic approaches to understand how the ovary develops over the lifespan. When we realized that we were unable to capture certain time points in ovarian development with human tissue, we formed collaborations with Oregon Health Sciences University and specifically the Oregon National Primate Research Center to start filling in gaps in primate ovarian development, the early stages of development, and the formation of the ovary in the third trimester, which is when follicles form, and follicles drive the function of the ovary. We've been doing spatial transcriptomics to map the lineages that form in the ovary over the ovarian life course. And through all of this, we've been making human pluripotent stem cells from women of known reproductive status. So as we're modeling the development of the ovary in the lab, we're using and beginning with human pluripotent stem cells that are known to make functional ovaries in women who have donated these cells for research. And with our computational work there, we've started collaborations with UCSF and also with Harvard University and Mass General Hospital. So in building cell types in the lab, going back to, we need to understand how cell types are built in the body. And so we've been using a lot of transcriptomic approaches in order to identify the stages that are necessary to build those cells in the body. And what we've been doing recently is a lot of spatial transcriptomics in order to understand not only how the cell identity, the transcriptional identity of these cells, but also where these cells live in time and space within the ovary. And so this is just some examples of the way we've been using spatial transcriptomic technology in order to map ovarian development in vivo in the body. What I want to point out in the slide on the right, it's my right, it's your right as well, is that each one of those dots is actually a computational dot. That dot is identifying a cell type in the ovary, either a granulosa cell, a germline, a progenitor, or a stromal cell. And what lives underneath every one of those dots is 1,000 genes. So what we're able to do is not only identify all of these different cell types within the ovary, but also a pretty good transcriptional signature of what's found in all of these different cell types in their spatial contexts. And we use this information to build our models in the lab. So what do our models in the lab look like? So on the left is the proof of concept that you can build an ovary entirely in vitro in the lab using stem cells. And so this is in the mouse. This is where the proof of technology lives, and we're translating it now to building ovaries in the lab using human cells. So in the top, what you're looking at is a little ovarian organoid. The germline is shown in blue. They're the oocytes, and in gray are the somatic cells that are supporting the oocytes. These are called granulosa cells. These are the hormone-producing cells of the ovary. In the middle, in the merged DAPI, that's what the cells look like within this reconstituted ovary. So you can see that we combine together single cells in the lab, and they self-organize in order to form the ovary, and they form little follicles composed of an oocyte, the granulosa cells, and the stromal cells. What we've been innovating on top of this technology is figuring out, can we generate a hormone-producing organ in vitro? Because if we want to develop new non-hormonal contraceptives, we need to not fiddle with the hormones of the ovary. And so we've started to work on those technologies in order to build these organs in the lab that produce hormones in a cocktail of medias that we've generated. And these follicle cells will progress with their gene transcriptome as well. And in the bottom in the middle is just demonstrating that you can make estrogen, progesterone, and testosterone within the ovary. So yes, the ovary does make testosterone as well, if you didn't know that. So now that we can generate ovaries in the lab from mouse, we generate ovaries in the lab, we're generating the cell types, I should say, of the ovaries in the lab from human, using our human pluripotent stem cell lines that have been consented to research of women from known reproductive status. For many years, my lab has been interested in the germline. We make germline cells in the lab in vitro routinely. We're moving into two-dimensional platforms to be able to manufacture germ cells from all pluripotent stem cells and not very small populations. So this is just an image of some of the germline that we make in the lab in 2D platforms. We make our ovarian somatic cells in 3D platforms from human pluripotent stem cells. And in blue, what you're looking at is the early progenitors of the ovary that we're able to make within these ovarian organoids without a germline. So the next step we're doing is combining them together to make the ovarian organoids so that they look like the image on the left, which is a reconstituted ovary. So in addition to building ovaries in the lab, which is for drug discovery, which is for safety screening, toxicology screening, that's the reason why we make these ovaries in the lab. We've also become very interested in the ovaries during a time in life that has not really been evaluated. So as Yalda said, the placenta also has unknown biology. The other area of the ovarian biology that is not well understood is the midlife period of a woman's life. So this is the ovary between the ages of 40 and 60. So this is when women tend to have their ovaries removed. They're thought of as a vestige, an organ that has no function anymore because it has no oocytes in it and it has no granulosa cells and it doesn't make estrogen or progesterone. And so that's why women go on hormone replacement therapy in order to improve their quality of life as they move through this menopausal transition. But I think that we owe women more than just hormone replacement therapy. Why aren't we focusing on rejuvenating the ovary so that the ovary itself is able to make its own estrogen and progesterone so women do not have to rely on synthetic hormones, which also have their own side effects as well? So what we thought we would do is let's look at a midlife ovary, an ovary in a woman from 40 to 60. Because as it turns out in the scientific literature, no one's looked. That's why it's removed. So what we've discovered is the midlife ovary is extremely cellular. So unlike the postmenopausal ovary, which is very fibrotic, this midlife ovary has a lot of cells in it. So the question is, what are those cells? What do they represent? How can we target them so that we can actually rejuvenate them so we can begin to kickstart hormone production again in the ovary without an oocyte? So that way, what we'd be building is an ovary that has hormone function, but not reproductive function. And that begins with understanding what the biology of this ovary is. And so we do this work in collaboration with Atlas Precision Health, Professor Yuna Kang, who's a pathologist at UCLA, as well as my colleague Sanaz Namazadeh, who's an MD PhD in the Department of OBGYN, and our commitment to improving women's health through a focus on the ovary. And with that, I'd like to thank you very much.
Thanks so much. When I was in medical school, we thought everything that had to do with being female or a woman was because of estrogen, and males was testosterone. We now know that the chromosomes, XX for women, or females, I should say, XY for males, matter. And I want to introduce you to Dr. Karen Rui. I'm paraphrasing, but my favorite title for a talk of hers was something like, "Do my 2X chromosomes make me look fat?" So I'm going to turn it over to Dr. Rui.
All right, thank you very much. Good afternoon. Yes, I'm going to tell you a little bit about our studies to understand mechanisms that underlie sex differences. And we're particularly interested in cardiometabolism. So first, I just want to illustrate how different the metabolite levels are between a healthy young man and woman. You can see the distributions here. This is typically not taken into account too much clinically, but we know it's important, and we know it probably reflects many of the cardiometabolic traits that we see that we know are different between men and women. This includes body composition, where mammals typically have more fat storage than males, all the way up to disease states such as stroke and heart attack, and even response to drugs. So we know that one of the key sex determinants of these, of course, are the hormones produced by the gonads. But as Janet was just alluding to, we're also very interested in the genetic sex. How do the sex chromosomes influence this? We know they're very important for development of the gonads, but what we've learned over the past several years is that they actually act in virtually any cell of the body, and they're active throughout our lifetimes. This is very important to understand. The challenge is, in a typical human or animal model, if an individual has XX chromosomes, these are typically paired with ovaries, and XY chromosomes are paired with testes. So how can we kind of disentangle this? So I was asked to tell you about one of the models that we used. It's known as the 4-core genotypes mouse model. And this model allows us to disentangle those two different components of biological sex. So we have 4 different genotypes that we study with this model. They include XX animals with ovaries and XY with testes, this typical genotypes we're used to, but also XY animals with ovaries and XX animals with testes. So this allows us to make comparisons among the 4 types and ask whether a trait we're interested in is determined by gonadal sex, where animals with ovaries differ from those with testes, or could it be influenced by chromosomal sex? XX animals differ from XY. And we've used this model to study many, many different traits. And it's actually been quite surprising to me how much of an influence the sex chromosomes have. So I mentioned that females typically store more fat mass. This is true in humans and all the way down to mice. And we see that actually, in addition to gonadal hormones, the sex chromosomes play an important role. Having XX chromosomes promotes fat storage on a high-fat diet compared to XY chromosomes, irrespective of what type of gonad is present. We also see the X chromosomes influence many traits, such as fatty liver, atherosclerosis, and even adverse drug effects. I'm going to tell you a little tiny bit about that. I'll just show you this figure here. Statin drugs are known to have adverse effects that are worse in females than in men. And we found using this model that if animals have an XY chromosome, whether they have ovaries or testes, they don't develop these adverse effects, one of which is glucose intolerance. But if they have 2X chromosomes, they do develop this. So seeing these sex chromosome effects, especially the influence of 2X chromosomes, has led us to try to determine why that is. So one thing we know that's very special about cells with 2X chromosomes is that one of the X chromosomes is inactivated early in development. And this actually normalizes the dosage of most of the genes on the X chromosome between an XX and an XY cell. However, you might not be aware that there are specific genes on that inactivated X that are still transcribed and expressed. And those genes are very particular. They're conserved between mice and humans, and they are always the same genes. So we have knowing that those are specific genes that escape inactivation, we propose that maybe some of those are influencing the effects we see that differ between XX and XY animals. We've tested various candidates, and I'll just show you a couple of examples here. One of these genes is known as KDM5C. It's a gene that regulates gene expression of other genes across the whole genome. We found that it associates with this increased fat composition in XX animals. So if we now then go in and modify not the whole chromosome dosage, but just the dosage of this one X chromosome gene, and take it down from the 2 copies normally present in a female to 1 copy, which would typically be found in an XY cell, or even no copies in the fat tissue, we find as we reduce that copy number, we reduce the fat, and we actually change the composition, the characteristics of the fat. We go from a typical nice white adipose tissue where you can see those beautiful cells filled with white fat, that's lipid, to a type of adipose tissue that actually has more of a role in using energy than storing it, and just by modulating this one gene. And this particular type of fat increases the energy expenditure in the animal, and we think that that's one of the reasons that they have this reduced fat. We can also do a similar analysis for other traits. I mentioned the statin adverse effects. And on the left there, you see a graph showing that in women compared to men, adverse effects from statin, including myopathy, as well as statin-induced diabetes, are more prevalent in women. And people have no idea what the mechanisms for this sex difference are. So we utilized our 4-core genotypes mouse to investigate this. As I showed you earlier, we found that XX chromosome dosage is promoting these adverse effects. And again, we tested candidate genes from the X chromosome that escape inactivation and found that this same gene, KDM5C, is one of the key players. So if we look at an animal, a standard female animal, she'll have 2 copies of KDM5C along with her XX chromosomes and ovaries. And in the red bars there, when you give this animal statins, she develops glucose intolerance over a very short time. However, if we now reduce the copy number down to 1 copy, as is normally present in a male, we can see we can prevent that. And it's our opinion that if we understand the targets of these genes, such as KDM5C, we can now identify new targets that might be important for intervening in the health of females versus males. And so if I can leave you with a couple of implications of this kind of work, I hope that it leaves you with the idea that in addition to gonadal sex, our chromosomal sex is very important in influencing many different traits. We study mostly metabolic traits, but this has been demonstrated for things including Alzheimer's disease and so on. And in our work, we've shown this is partly mediated by these special genes on the X chromosome that escape inactivation, and therefore they're always going to be present at higher levels in an XX cell and an XX organ and an XX animal or human than in an XY. And the way that these function is quite exciting. The ones that we have focused on that have big effects, they themselves regulate gene expression across the genome. And so if we can identify those targets, we can identify further into the mechanism. We also have inhibitors of the action of some of these. And we find that when we use those, we can ameliorate some of the effects of having those dosage differences. And then so finally, when you think about our lifespan, of course, we have a dynamic level of gonadal hormones throughout our life. In men, for example, testosterone levels start to diminish at around age 30. And of course, we know menopause, age 50 and beyond. And so the effects of those gonadal hormones is changing throughout our life. However, the sex chromosomes are present throughout, both early and late when the gonadal hormone levels are low. And therefore, we find that at those periods in life, these sex chromosome dosages really become important in influencing these traits. Given that we have so many chronic diseases that occur later in life, we think that these might provide important targets for some of those diseases. That's it. Thanks for listening.
All right. And finally, I'm really excited to introduce Dr. Carol Watson. I remember when I came to her for mentorship about this topic in terms of research. And I still remember we were sitting in the cafeteria and she looked at me and she said, Janet, in research, even the rats are male. So I'm going to turn it over to Dr. Watson.
Thank you so much. I'm going to talk about our work in the Women's Cardiovascular Center. We are dedicated to prevent preventable diseases. And one of the things we look at a lot is sort of the low-hanging fruit. There's so many things that happen every day that we're exposed to every day that have big impacts, and that if we can get those under control, we think we can improve women's lives. And it's important to remember that heart disease is the leading cause of death for women. It's been that way ever since we started keeping records. So you can see heart disease is number 1, cancer is number 2. Many women believe that breast cancer is their leading cause of death. But as you see, it's a very important pathology, but more than 13 times more women each year will die from heart disease than from breast cancer. The other thing that's really distressing is that heart disease rates are increasing. This is data from 1900, like since we started keeping records. And crazy thing is that for years, heart disease rates were just exploding rapidly because we were doing everything wrong, even though we didn't realize it. We were eating meat and potatoes because we thought it made us strong. We were smoking because it thought it settled us down. We weren't doing things that we know now are healthy. But decades of research have shown us that the risk factors like high blood pressure, we need to treat, high cholesterol, we need to eat more healthily, we need to watch our weight. And when we did that, we saw heart disease rates start to stabilize and, in fact, start to go down. The sad part and the scary part for us is that since 2015, heart disease rates have gone up, both for men and for women. And we don't know all the reasons why, but we're clearly doing things wrong. So we're trying to figure out in our lab, what are those things? One of the craziest things is that even amongst our youngest women, heart attack rates are increasing. That pink line there is the heart attack spike in young women. And ever since the COVID pandemic, there's been a heart attack spike in both men and women, but young women especially seem to bear the brunt. Now, we do a lot of epidemiologic research. The multi-ethnic study of atherosclerosis is a large NIH-sponsored epidemiologic trial. It's looking at the correlates and the causes of atherosclerosis. So, in the 1950s, we had the Framingham Heart Study that told us that high blood pressure was bad, meat and potatoes was bad, diabetes was bad. That was great, but it was done in a small town in Massachusetts, which was carried out mostly among men. And so it was time to update it. And so the multi-ethnic study of atherosclerosis is that new study. It's a quarter Black, a quarter white, a quarter Chinese, a quarter Hispanic, and it's going to try to tell us what are the new risk factors for heart disease. Why are we going the wrong direction? One of the studies we did in Mesa was to look at men and women and bring them into our center, ask a bunch of questions, figure out what their blood pressure was, what their cholesterol was, what their glucose was, all of those things. And we looked at the differences between men and women. What we found was that women had a number of risk factors that were related to cardiometabolic risks, the things that you just heard about, a lot more obesity, more hypertension, things like that. But the other thing we were interested in was looking at stress. Now, you can look at stress by asking people, like, how stressed do you feel? And that can give you one answer. But you can also look at the physiology of stress. So we actually asked people to collect their 24-hour urine, and we measured their stress hormones. So whether you feel like you're under stress or not, your body knows. And if you're under stress, your body will be surging with these stress hormones. What we found was that on any random day, women had more stress hormones surging than did men. They had more norepinephrine and more dopamine. They had similar epinephrine and cortisol levels, but the overall risk profile was that these women had more stress hormones. So we figured we need to figure out a way to get women to have more risk factor modification to improve their risk factors, and also to try to get them to have less stress. So what do we do? By the way, if you have more stress hormones surging in your body, you're going to have more heart disease. That we've shown. Okay, so what we did was to try to get people to adopt healthier lifestyles. We did a study called the ECO study, the Early Cardiovascular Health Outcome Study. We said, now we know our female patients, and most of them are rule followers. They know what they should be doing, and they want to do the right thing. But things like life gets in the way, picking up the kids or working on their advancing their career, things like that. So we said, what if we can do something just to give them simple reminders about heart healthy behaviors? And we decided to use a text messaging platform using nudge theory. So nudge theory, positive reinforcement, indirect suggestion. So we'd say, did you know people who sleep between 7 and 8 hours a night live longer? And we didn't say, you got to get to bed. We just said, hey, did you know? And we want to see, does that going to translate into them sleeping more? We said, did you know that one of the highest sources of salt in the diet is baked goods? Things like that. And so we saw that by giving these little nudges, we actually saw a reduction in unhealthy behaviors, a better risk factor profile. Now, when we wanted to get women to reduce their stress, we said, well, we can ask them to do meditation or mindfulness or things like that. But we heard over and over again from women that, hey, I just can't seem to, quote unquote, get in the zone. So we wanted to figure out a way to help them get in the zone. And we realized that maybe virtual reality would be a way to do that. So we did a study looking at a novel virtual reality experience for stress reduction in our patients. Now, this is a virtual reality experience that was just a bunch of geometric shapes and calming music. It wasn't pictures of things. And the idea was to help them get into the zone. Now, and we didn't want to just know if they felt less stress. We wanted to see physiologic responses that correlated with less stress. So one of those things is heart rate variability. Your heart should not be like a metronome. It should not be boom, boom, boom. It should be variating, boom, 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 boom. It should change. And the healthier your heart is, the more heart rate variability there is. So we did a heart rate variability index, which could tell us how much variability there was in the individual's heart rate. The lower the number indicated greater heart rate variability. And so the greater the heart rate variability, the healthier the heart. It indicates a healthier balance between the sympathetic and the parasympathetic tone. So what you can see, this is just the example of our data from the virtual reality experience. Within 5 minutes, the heart rate variability significantly increased, and it stayed up for the whole duration of the virtual reality experience. It was very, very enjoyable. People said they really enjoyed it. They got in the zone. They felt their stress relieved, and we could see physiologic mechanism and parameters that indicated their stress was being reduced. So that's one of our uses now, virtual reality. We have a second study that we're trying to start now. One of the things that we've seen epidemiologically is that regular exercise is good for everyone. It seems to be maybe have an even better impact on women than men. But women tend to engage in less leisure time physical activity than do men. And a lot of women just don't see themselves as athletes. So we're trying to start a virtual reality program where we actually have them showing them doing games, exercising, playing tennis, playing pickleball, etc., to get them in the idea that, yes, you can exercise. So we'll let you know next time how that works out. And thanks so much for your attention.
All right. Thanks so much to everyone. Any questions or comments from the audience? Yes.
I know that from what you said, that heart rate variability is indicative of good heart health, but is it also from the study you showed, is it indicative of the stress? Is it correlated with the amount of stress that a woman has?
So we've never looked at that specifically, but what we know is that the healthiest hearts have a lot of heart rate variability. And if you have high stress, you tend to have higher sympathetic tone, and higher sympathetic tone is correlated with lower heart rate variability. So we would expect that to be true, but I don't have that data.
Yes.
This is maybe a very general question, but as you look into the future of women's health, what are you most excited about? I think there's a lot of sort of, maybe not negative, but we all have lots of thoughts, but what are you most excited about?
I'm excited about the $2 billion of funding we hopefully will get.
So I'm really interested in the increase in attention on women's health and women's disease. I think for far too long, as Yalda said, the women's body has been underrepresented in biomedical research. And so what that means is as fewer and fewer labs building technologies that can help women, that means that although there's a really robust public health pipeline of improving equity and access to existing reproductive therapies, and I mean reproductive generally, so access to maternal care and health. There's fewer and fewer innovations that are more upstream, so fewer scientists working in the field, just fewer. And so I think we're doing a real disservice to women by not increasing the pipeline of training and creating opportunities for young scientists to work in the area of women's health. So that funding is particularly important to attracting people to the field because they can see that there's a viable option now to starting a lab and building technologies that are going to help women. What I see as a scientist, I also teach in the undergraduate campus. There's a real interest in women's health and in reproductive health amongst people who are 18 to 25. And they're hungry for information and they're hungry for opportunities. And so what excites me is that there's a lot of excitement in the field right now. And I'm expecting that this is going to change into solutions for women.
All right, I'm going to jump in. I think it's the first stage in true precision medicine is to understand just men and women, right? So I think all of these things are key to the point where we can generalize personalized medicine beyond a few specific diseases.
I study pregnancy, but I think it's time to stop thinking women's health is, women and children. Women and children hospitals. Women are through a life course. And when we think reproduction is a tiny, tiny, tiny segment. And those 40 weeks are nothing in comparison to the 80, 90, 100 years that they live. So just the attention that women's health is getting that goes beyond being a womb, even though that's what I study, is absolutely exciting. And to think of it that we're openly having these discussions, that there's now, tampons and pads in bathrooms at UCLA, and it's part of people's advocacy. And we talk about choice and reproductive life planning. So that's all been really, really exciting.
And I wanted to follow up that with one other comment, which is that I don't know if you are watching TV and you see some of these ads for actual medical delivery systems that are catering to women. And I think that's sort of reflective of a recognition that women actually are or more than half of the population. And we are a consumer force. And I think we need to exert that consumer force. And so even though there is, there's $200 million going to, new grants, that's what goes to prostate cancer in a year. So it's good. It's a start, but I think some of this needs to come from us as consumers, from the community in general, that women aren't going to take being handed the one size fits all medicine anymore. And I think that push kind of from the commercial side of things with services that are aimed at women, particularly, I think, again, I would echo tremendously that older women are a very much ignored portion of the population, but they've got some money and they should use it and exert that force in our economic system to really demand better treatment, better care, individualized care, and acknowledgement that these are real issues that women face and that there are solutions for if we work at it.
I think we've got in the back.
Do you believe that, based on the studies of the 70 kilogram man, that prescription drugs are all for men and they're not really catered to women's bodies?
Yeah, I'm going to let, Carol obviously is very expert in this, but I'm just going to comment. I lecture on this topic, and all I have to do is go to the latest New England Journal of Medicine, right? I just have to look at one copy of the New England Journal of Medicine to find a drug study where over 70% of the subjects are males. And in fact, there was a recent study published looking at prevention of a sexually transmitted infection, okay? This is a sexually transmitted infection that if you are a male, it's mildly irritating. If you are a woman, it can literally be fatal. Okay, this was, prevention of chlamydia and gonorrhea. 95% of the people in this study were male. So yes, we need to keep working on this. Other comments?
That's about as good as the study for a female sexual dysfunction drug that was tested entirely in men.
Yeah, I mean, when we studied hormone replacement therapy in the 60s to see if it would reduce heart disease, it was done all in men, estrogen in men. So it's crazy. But all you need to do is look at the current adverse drug reaction database. 90% of the drugs that are withdrawn due to adverse events, the overwhelming adverse events are in women. They're taken off the market because they're actually killing women. Who do we know? It might just be a dosing issue. We don't know. I am a cardiologist. I treat a lot of patients with statins. I was actually on the National Guideline Writing Committee about statins and statin dosing. I understand that 80 milligrams of Lipitor is the right dose according to the clinical trials. But I can tell you, women have many more side effects to statins, and those who have the most adverse events, side effects to statins, have the strongest response. So we can get away with alternate day dosing, cutting the dose in half, things like that, because all of the efficacies related to muscle mass in women tend to have less than men.
Yeah, and I think that gets back to, again, the initiative that Dr. Watson was there when it was announced, which is really looking at what are the rules currently. So as an example, within the past few years, the FDA actually approved a drug to prevent the acquisition of HIV only in men and transgender women. And the question is, should the FDA be allowed to approve drugs without studies in individuals with an XX chromosomal complement? So I think that's just one of the topics. I think we have time for one more question. Oh, I'm going to have to pick. Yes, I saw you first.
Thank you for a very engaging discussion. I just wanted to know how everything you said is extremely meaningful. How can we have more impact? Can you write all of that, what you just said, in a blog or a monthly manuscript or whatever that will increase awareness for women all ages, not just young, everybody? Because I think in general, every statement you mentioned has such a big impact on all of us. But we tend to kind of read New England Journal of Medicine, we read, JAMA, etc., but we don't capture what you just said in this just 1 hour, and so whatever time. Have you thought of putting together, as women in this advocates for the rest of the women population in the United States and the world?
Well, I think we're almost out of time, but I think I want to give Dr. Watson the last word on that, because she's been so involved with the American Heart Association and how advocacy has been done.
Yes. So the answer is, specifically for us, no, we haven't thought about that, but we really should, you guys. Okay. But I think what we've done is, when you look at the reduction in heart disease deaths amongst women, nothing moved the needle until we started large public awareness campaigns. That's what moves the needle. When we tried to talk to providers or regulatory agencies, nothing happened. But when we started going to women saying, guess what, you're at risk, and that started changing things. So we need to. So
that's a tremendous last question and last word. Thank you to the audience and to our tremendous panel.